All right, so this should be it. This should be the last mini lecture of the year of the textbook because we are at the end of the textbook. And remember that we introduced this idea that protein synthesis is carried out in 5 stages. We talked a little bit about what the ribosome looks like. We talked about how we'll go through each of the stages. So let's start with stage 1, the activation of amino acids, because we've talked about the old evidence that it happens, but what does it actually look like? Well, for one thing, you need to say, what does a tRNA look like? Because we're talking about attaching the amino acids to this. This is the secondary structure of a typical tRNA. It has a clover leaf sort of structure. And see at the bottom you have the anticodon. That's the part we've talked about before. The other parts in pink are distinctive, unusual, they're modified nucleotides in some way. And you see there's a lot of them. There's a lot of places where this has been modified post-translationally, or post-transcriptionally, I suppose, where you have some kind of chemical group put on so that it's a unique chemical binding site. And you know that this is, number one, it's a tRNA that it knows, but it also is the tRNA that is coding for whatever the amino acid is. In this case, the amino acid that's coding for needs to be attached to the 3' end. And that's where you always have this CCA sequence, which is what the enzyme that loads the amino acid up will bind to and attach the amino acid to. This is what, so this is another way of saying, this is coloring the different kinds of modifications that you find in tRNA. 10% of the nucleotides have some kind of modification. So this thing is really tricked out. It's kind of like your friend who has all the tattoos, right? They've had the modifications to their own body that they put on, and they just love to get tattoos. This phenylalanine tRNA loves to get the cellular version of tattoos, which might be like a methylation or a deamination or something like that. By the way, the A to I enzyme, the ADAR enzyme that we mentioned, that is the kind of thing that we're talking about when we say modifications. But of course, that's a secondary structure. What does it look like when it actually folds up? So those two arms sort of come together, and it actually makes an L-shaped structure with the anticodon at the bottom and the amino acid arm at the top. So you can see how that sort of folds up, and it makes this distinctive L. So there's a CCA, and in this case, this is the phenylalanine. The phenylalanine is attached to that purple region up there. The anticodon, we know that the anticodon for this particular one is AAG, and I'm reading it, actually I'm reading it backwards, I flipped it around. It's actually GAA, but remember, phenylalanine was UUUUU, right? So it makes sense that we would expect AAA to be one of the options, but remember that third base, we want to be able to bind other codons. The third base of the codon, the first base of the anticodon, is actually different. And I believe that follows the wobble rules that we mentioned. So the thing is, you've got the specific tRNA, the specific shape, you got the specific amino acid, you need to get the two of them together. So you need the enzyme that's going to form the ester bond, it's going to match make the two. And remember that sometimes you have 2 or more tRNA molecules that correspond to a particular amino acid. That's all worked out because the binding sites for those will match up on the particular synthetase. That way we only need 20 of them. It's pretty easy to have an overlapping kind of binding site like that. Technically the word for that would be a degenerate binding site between the two tRNAs. So the same enzyme usually will aminoacylate all of the tRNAs for it, and there ends up being 20 different aminoacyl-tRNA synthetases. You can't get below 20 because you need 1 for each amino acid. An ester linkage will actually be, when you hydrolyze it, you actually end up with a more stable molecule. So there's a large negative delta G. When you have formed it, that means there's a large positive delta G to form it in the first place. When you break it, when you have formed it and you break it, I didn't finish that sentence, you get negative 29 out. That means it takes 29 at least to make it in the first place. So how does that happen? It's an enzyme, so let's talk about it. Again, it's phosphate chemistry. This one's a little more interesting than most of the other phosphate chemistry we see. You take the amino acid, the carboxyl group of it will attack the alpha phosphate. You're used to this by now. This means you'll end up adenylating the amino acid. and you end up with the PPI going off to help drive the reaction forward yet again. So the adenylated amino acid will actually be the substrate for the synthetase. The synthetase will take this, which has an acyl phosphate, which has a lot of energy in itself, but because it's synthetase, you can see that ATP is also involved, and then it's made. And you notice that there are two branching paths, and I'm not showing you any more of that. Basically, you have this highly energetic molecule, and you use this to actually put it onto you trade this for the ester bond. This acyl phosphate is like negative 45 hydrolysis energy, and so it's pretty easy to make this into an ester bond with driving force left over for the reaction. The end result is that you form this ester linkage. It's about negative 30 hydrolysis, so you've gone from negative 45 to negative 30. That means you have about 25 or 15, I'm sorry, of 15 kilojoules per mole of forward driving equilibrium power to produce more products and reactants. So this is what it looks like, the ester linkage. And so it stands to reason, if we understand how this all works, maybe we can use this for ourselves. And there is a particular lab, and I believe this is being done by a lot more people now. For a couple decades, it was only the Schultz lab at Scripps that was able to make it work for us to modify the tRNA system to be able to reprogram codons to do different amino acids. So instead of phenylalanine, you have this ketone, this phenyl ketone. I wish I had the names of these actually, because I'm sure they have cool names. But just look at these. You should be remembering what the amino acid structures and labels are. That's pretty fundamental biochemistry that we've been using all through the class. Hopefully you can look at these and see these are not normal amino acids. These are organic chemistry made these amino acids. The Schultz lab and those who followed their techniques can put them onto specific tRNAs and have them be put on by the ribosome when they have a specific codon that shows up in the modified organism that does this. But of course, to do this, they don't just need a new tRNA, they need a new aminoacyl synthetase that will go for the particular codon that they are trying to modify. Once it's all in place, they can actually take the codon, they can actually program DNA by just putting the codon in the DNA. They give it to this organism, and it will make proteins with this weird group put in wherever that codon says to put it in. Really cool science, but it's really complicated because you don't just need one thing. You need to change the whole system for the whole codon, which includes the aminoacyl synthetase. So this is one example. This is expanding the genetic code to include a 21st amino acid, phosphoserine, that will go for, you see it has the GCA anticodon, so that means it goes for a TGC. And so that particular codon, I believe that, I could look it up, but it would take me away from my screen. And so you see down here, TGC will actually bind. Oh, and if you look down here at the very bottom of the screen, they show you the tRNA. Look at this. They show you the codon. It's actually, so the way they're showing it, oh, and by the way, I was reversing it when I shouldn't because I got to remember the 3' end is up here. And so you actually end up with these two bases in the codon binding this way. So they will bind GC codons. And they call it tRNA sep. Look at how they have it down here with the little sep. That means it's tRNA that targets the sep codon. Phosphoserine is what they're calling it, SEP instead of SER. And so they need to make the sep-RS enzyme that would be able to take the sep and put it onto the special tRNA. And so you see up here, the regular tRNA usually encodes for cysteine, but they're taking that exact codon and they're making that codon put in a phosphoserine instead. They're deliberately doing this because every codon in that organism that originally coded for cysteine of that particular codon will now make sep in its place. And so this causes other problems for the organism. So again, you see how this is tricky. But the sep will actually go into the place and you change it. You basically have the CUA that binds the TAG codon in DNA. Yeah, I think I misinterpreted this when I was saying GC. CUA binds TAG. I was right in the first place. I should trust myself when I do these things. So they needed to redesign. They found that it actually wouldn't work until they designed stuff on the other end. When we talk about elongation factors, they had to redesign that as well to make the system work. But the good news is, when you put in all this work, it actually will make a living organism that will use a 21st amino acid based on a recoded codon. You have redesigned the entire system from codon to protein result to use a new amino acid. And so now they even have facilities for this. This is a core facility at Oregon State that will make unnatural protein, probably because it's too hard for individual labs to do it themselves, but it's useful. And so they actually will have a facility of technicians and scientists who do this all the time, and they'll help you figure out how to use it for your lab. I just think that's a cool name, Unnatural Protein Facility. I'd kind of like to get a shirt for that. So the thing is, there's actually a proofreading step that happens between the attachment of the amino acid and its use on the ribosome. This is kind of cool, and we know it exists because we see how faithful the technique is. Like, let's say that the isoleucine synthetase. It must bind to isoleucine but not valine, and think about that. That's the difference of a single methyl group. And going one way, binding valine but not isoleucine, is easier because you just make a site that's smaller and it binds the smaller thing. But how do you bind the bigger thing but not the smaller thing, especially when you have this non-specific hydrophobic interaction that's the only thing that you've got to discriminate based on? So it turns out that you actually have the synthetase itself has a binding site that fits isoleucine, but it's only able to discriminate by a factor of 200. For every 200 isoleucines that it binds, a valine accidentally gets in. So it turns out that valine is there only in 1 out of 3,000 chances. So if you look at that, that means there must be a 1 in 15 kind of proofreading step that gives us the observed 1 out of 3,000 mistakes, when the enzyme itself only does 1 out of 200. So the difference is because there's a proofreading step, which is a different binding site. I believe this is on the same enzyme, but it's a different binding site that will bind valine AMP and hydrolyze it. So that's the way that you do it. You have a proofreading step that will destroy accidentally made valine so that you end up with the isoleucine is what persists. Very elegant solution, actually. So the one thing that we know is every aminoacyl-tRNA synthetase is going to bind only the tRNAs that it's supposed to. It's going to be specific for the tRNA or the 2 tRNAs that, or maybe even more, I don't know, that will code for that, it will bind all of the arginine synthetase will be able to bind all 6 arginine tRNAs. It should be able to do that. If you only have 20, it must be able to do that. There's some kind of structural code in the tRNAs where all the arginine tRNAs have a particular structure that enzyme binds to. And so we can look at this and we can try to figure out the code. For example, the blue positions are the same in all tRNAs. If you just want to bind any tRNA, you can go after those sites. But orange and green are the known recognition points, and those will differ between tRNAs. So the idea is that arginine enzyme will bind the 6 that have particular structures at particular points, and the other tRNAs will not. And so sometimes those specially modified bases we noticed provide the specificity that you need to be able to make this whole system work. For example, this is one, and they have a large binding area to work with. This is glutamine aminoacyl-tRNA synthetase, and it's binding its tRNA with a very large surface area. There's the tRNA. There's ATP, by the way, and there's a very large area where that is binding. So that is that shows you how it works. And if you have just the right binding sites, though, you can make a truncated RNA work. For alanine, they found out that this mini helix RNA apparently has what it needs to bind its own enzyme and to charge that up with alanine RNA. I think that's pretty crazy that the rest of it is not basically doing anything, but apparently that's how this tRNA works. If you just delete all the nucleotides between 13 and 66, it still works. Is this a clue for how it developed? Because it's much easier to imagine this evolving than the thing on the left. Who knows, but I know that it works, and that's kind of cool in itself. Okay, now I've got one more thing for you to watch. I'm going to have 2 videos posted about translation and action because it's easier for you to see them on your own computer than for me to show it on a screen. I'm going to ask you to look at those, and then you'll have it in your head when we talk about it on Monday. So thank you for observing these mini lectures. I will see you on Monday.